Hi everyone, my name is Honggu Lee. I'm from Harvard Mass General Hospital, and I would like to thank Brick for giving me such a great opportunity to present my work today. So today I'm going to talk about removing the root cause of the Fragile X syndrome via small molecules and loop formation. So autism spectrum disorder uh, uh, can be caused by many different reasons, and most of them involves very large chunk of the DNA depletion or um, replication. So usually many genes are involved in this causing this autism spectrum disorders. But in the case of the Fragile X syndrome, is caused by only one gene called FMR1. And this FMR1 gene is important for brain development. And all of us here has some length of the CGG repeat in this FMR1 5' UTR region. And most of us has less than 50 times of this CGG repeats. But once this CGG repeat gets longer and longer, and it becomes longer than 200 times, it causes complete epigenetic silencing of FMR1 gene. And this makes this FMR Fragile X syndrome patients suffer for intellectual disability and autistic behavior. In order to study this repeat-related disease, the autism spectrum disorder, Fragile X syndrome, I wanted to first monitor this repeat length. And as you can see here, in the case of wild-type iPS cells, this cell line had around 20 times of CGG repeat. And on the right lane, when I treat this sample with the restriction enzyme called HPA2, which cuts unmethylated DNA, then you can see this band disappears. So this means this wild-type cells has this CGG repeat that are not DNA methylated. On the other hand, these two cell lines that are derived from Fragile X patients, one is iPS cells and the other is human ESL. And those two cell lines shows much longer CGG repeat, longer than 300 times. And this signal doesn't go away after treating HPA2, indicating this long CGG repeat are heavily DNA methylated. So as you can imagine, this more than 300 CGG repeat is 100% GC with the longer than 1 KB. So this is really extremely hard to clone. So using the iPS cells or the patient-derived cell is the almost only way to study these long repeats. And due to this long repeat and DNA methylation, these two Fragile X lines shows very strong FMR1 silencing, as you can see. So there was almost no detectable level of the FMR1 mRNA. So I have been trying to reactivate these genes because if I can reactivate these genes, I can we can use it for the therapeutics for this Fragile X syndrome patients. So I have been trying many different ways, but mostly I failed. But one method worked, which is called naive stem cell media. So what is naive stem cell media? So when we grow the human ES cells for a long time by cell culture system, they tend to lose their pluripotency because of the epigenetic environment changes. And people wanted to make this primed human ES cell back to the naive human ES cells, which is closer to the inner cell mass, which is the origin of the this human ES cells. So people try to come up with the method to reverse this epigenetic state by using many different recipes. And I tried two of these naive stem cell media, one called Reset and the other called 5I, which means 5 different inhibitors, as you can see here. So unfortunately, when I treated Reset, it didn't show any significant increase of the FMR1 gene reactivation. And I forgot to mention that this naive stem cell media, one of the effect is the global DNA demethylation, which is one of the steps that can reverse this epigenetic state from prime state to the naive state media, to the naive state. So when I treat the 5i media for 12 days, I was able to see very strong reactivation of FMR1 gene, not only the mRNA level, but also I could see recovery of the protein level as well. One very striking observation was found from the long repeat. So as you can see, before treatment, this cell has around 300 times CGG repeat. And as I treat this 5i media longer and longer, this long repeat becomes shorter and shorter, and it ends up with this smear band with the heterogeneous shorter length of CGG repeats. On the right panel, I use the bioanalyzer to more properly quantify this length distribution. So if you see this signal from the long repeat, the day 0, this blue signal goes down and down and down. And the signal from the short repeat is increasing more and more as I treat 5i media longer. I also wanted to see whether this can be reproducible. So I used many different cell lines that are derived from the single cell. So they all started from the long CG repeat. And when I treat 5i for a long time, this long repeat becomes shorter. And I wanted to also double check whether this smear band is not artifact of the PCR. So if they are really representing heterogeneous population of the cells with the shorter CG repeats, if I clone the single cells from this population, I should have this cells that has shorter repeats and without DNA methylation. So, and that's what I observed. So after picking many single cell clones, I was able to see many clones that are shorter CG repeat and no DNA methylation. And this cell line also showed quite high level of FMR1 level compared to the wild type. And also protein level was recovered as well. As expected, this 5i treatment triggered very strong DNA demethylation. I tested by 2 different assays. First is MEDIP, and second is pyrosequencing. And as you can see, both methods showed very strong decrease of the DNA methylation, indicating 5i is causing this strong DNA demethylation. On the other hand, another repressive histone marks H3, histone H3 K9 di and trimethylation. In the case of trimethylation was very high before treatment, and it shows strong decrease, but not as strong as DNA methylation. And most importantly, when I checked day 6, there was more than half of the K9 trimethylation left, but this at day 6, this FMR1 level was already start to show significant increase, indicating DNA methylation is the main source of the gene silencing. So I wanted to know why and how 5i media can trigger this strong DNA demethylation. And when I checked these 2 DNA demethylase, TET1 and TET2, the mRNA level of those 2 DNA demethylase showed very strong upregulation after 5i treatment. Also, when I knocked down those 2 TET1 and TET2, I was able to see quite significant reduction of the DNA demethylation, as you can see here, indicating TET1 and TET2 are indeed the players that cause the DNA demethylation. And this FMR1 reactivation level was showing significant reduction after TET1 and TET2 knockdown. So based on this observation, I concluded that 5i media triggers TET1 and TET2 upregulation, and this causes global DNA demethylation, and then trigger FMR1 transcription. Next, I wanted to optimize this FMR1 reactivation condition by finding the optimal component of these 5 different inhibitors. So I tested many different combinations, starting from the only one inhibitor testing and two inhibitor combination and 3 and 4 combination. And as you can see, 2 inhibitors, BRAF inhibitor and MAP kinase inhibitor, P and S, those 2 inhibitor combination was sufficient to trigger very strong FMR1 re-expression. But when I used these 2 combination, then the cells were suffering a lot. So I had to add third one, ROCK inhibitor, which is known to be helpful for maintaining cells in the stress condition. So when I used these 3 inhibitors, I was able to grow the cells and also see the very strong FMR1 reactivation level. And these 3 inhibitor combination also caused as strong DNA demethylation as 5i. And as a next step, since this Fragile X syndrome is an autism spectrum disorder, I wanted to test whether this DNA demethylation at the neuronal progenitor cells also can cause FMR1 reactivation and seizure repeat contraction. And indeed, when I treat decitabine, which is a DNMT1 inhibitor, I was able to see very strong reactivation of the FMR1 mRNA level. And also, I was able to see long seizure repeat contraction as well. So far, I have been trying to trigger global DNA demethylation. And I was curious, what if I can remove this DNA methylation at FMR1 specifically? So fortunately, from 2018, there was a very nice paper describing recruitment of the TET1 to the specific locus by using guide RNA with the dead Cas9. So this Cas9 cannot cut, but it can recruit specific enzyme by tethering by using the site-specific guide RNA. So by using this system, I was able to achieve site-specific DNA demethylation. And as expected, I was able to see very strong FMR1 reactivation after recruiting that Cas9 tether to TET1, as you can see in these red bars. And I was also able to see very strong seizure repeat contraction. So this was very nice data because I really wanted to see this seizure repeat contraction was indeed because of the DNA demethylation at FMR1 region. But very unexpectedly, when I used the TET1 dead version that cannot DNA demethylate as a negative control by tethering to the dead Cas9, I was able to see much weaker but quite significant level of FMR1 reactivation. And also, I was able to see quite strong seizure repeat contraction as well. So I was very perplexed because this was totally unexpected result. I expected no effect at all, but even using the catalytically dead TET1, I was seeing these 2 phenomena. So I was keep thinking what's going on and how can I explain this? And then finally, I came up with this idea that within the catalytically dead Cas9, there's a secondary structure that can be formed called R-loop. R-loop is a DNA and RNA hybrid. As you can see, inside of the dead Cas9, guide RNA anneals to the DNA template, and this forms very stable non-canonical nucleic acid structure, so-called R-loop. And this R-loop has been known to cause genome instability if it's not properly regulated. So I thought maybe this R-loop formation by the dead Cas9, even with this negative control, might cause this repeat contraction. So if that's the case, I also thought maybe when I use the 5i, this nascent RNA produced by Pol2 after DNA demethylation, this also can form R-loop, and this would also cause this CGRP contraction. So I wanted to test this possibility. I also want to explain that once R-loop forms, there's a single-stranded DNA part that can form secondary structure. And this single-strand DNA is a CGG repeat region. So this G-rich region is also very well known to form very stable secondary structure so that it can trigger DNA repair pathway. So in order to check whether this hypothesis is correct, I tested whether this R-loop has been formed in 5i treated cells. And indeed, I was able to see very strong R-loop formation in these 5i treated cells at FMR1 region. So, and if my hypothesis that this R-loop is causing this CG repeat contraction, what if I remove this R-loop after 5i treatment? So I tried this antisense oligo of FMR1 that specifically degrades the nascent RNA of FMR1 after 5i treatment. So after this ASO treatment, I was able to see this R-loop level decrease, and I saw this suppression of the CGRP contraction. So this indicates that R-loop formation was indeed the cause of this CGRP contraction. So far, I have been shown that 5i treatment upregulate TET1 and 2 that cause DNA demethylation, and that Cas9 with the TET1 tether also can trigger DNA demethylation at FMR1 and trigger FMR1 transcription, including the CG repeat region, which is at the 5' UTR. And this transcription can trigger R-loop formation, and this R-loop formation was sufficient to trigger CG repeat contraction. So next, I wanted to test what if without 5i, what if I induce the R-loop by using that Cas9? So this time I used that Cas9 that cannot cut, and it doesn't have any other tethered domain, like without that domain. And when I recruit this that Cas9 to the CG repeat region at FMR1, I was able to see strong increase of the R-loop levels. As a negative control, I tethered RNase H, which cuts the R-loop. So as you can see here, even if I recruit the dead Cas9, this RNase H tethered version shows decreased R-loop levels, indicating this signal is specific to R-loop formation by dead Cas9. And as expected, when I recruit the dead Cas9 to the FMR1 region, I was able to see very strong CG repeat contraction. Also, I was able to see much stronger FMR1 mRNA level reactivation by these dead Cas9 recruited cells. And unexpectedly, I also observed much stronger DNA demethylation when I recruit that Cas9. So what does it mean? So this was also unexpected, and I wanted to test whether this R-loop formation can trigger active DNA demethylation by recruiting TET enzyme. In fact, there was a paper from 2019 that described one of the long non-coding RNA from very different locus can form R loop, and then this R loop can trigger this TET1 recruitment and also DNA demethylation at that locus. So I wanted to test whether this can also happen in this Fragile X FMR1 region when I recruit that Cas9. So as you can see here, once the DNA is methylated, if this methylated DNA get by TET enzyme by demethylase, then it become 5-HMC temporarily, and then it become regular cytosine. So if TET enzyme removes this DNA methylation, there will be 5-HMC transiently. So I tested whether this 5-HMC level was increasing after that Cas9 recruitment. And indeed, that was the case. So I was able to see this increase of the 5-HMC level, indicating this active DNA deventilation is happening in this condition. So based on this, I was able to say that R loop formation itself also can facilitate DNA demethylation, which forms positive feedback loop here. So once R loop is formed, it can trigger more DNA demethylation, and DNA demethylation will trigger FMR1 transcription, and this transcription can form more R loop. And this positive feedback loop can start from 5i or that Cas9 mediated R loop formation. Next, since I have been using this guide RNA for the CGG repeat for recruiting that Cas9, I wanted to test whether I can use FMR1 specific guide RNA. In fact, there are so many CGG repeats in the genome, but it's much shorter than this Fragile X CGG repeat. But this guide RNA can cause non-specific dead Cas9 recruitment to many sites of the genome because of the CGG repeats. So I wanted to test this guide RNA that are unique to FMR1. Some of them are 100% unique region. Some of them were overlapping with the CGG repeat region. And among those, the guide RNA that I tested, one of them worked very well for FMR1 reactivation. As you can see here, this guide RNA shows as strong FMR1 reactivation as the CGG guide RNA. Also, this FMR1 specific guide RNA triggered strong CGG repeat contraction and strong DNA demethylation. Finally, I wanted to test whether this FMR1 specific guide RNA mediated R loop formation was specific to FMR1 by performing this RNA-seq. And as you can see, I only saw 2 or 3 differentially expressed genes, and one was FMR1, which shows very strong upregulation. And the other 2 was showing around 2-fold increase, and it was barely significant. So, so this shows that by using this FMR1 specific guide RNA, we can achieve very, very specific FMR1 reactivation and CGG repeat contraction. As a next step, I wanted to know how this R loop formation can trigger this CGG repeat contraction. Would there be the DNA repair mechanism involved so that I can see this gamma H2AX? And that was the case. So when I checked this DNA damage mark gamma H2AX, I was able to see quite significant level of this gamma H2AX after 5i treatment. And I wanted to know what type of the DNA repair pathways are involved in. So I tested a lot of different DNA repair pathways. And today I'm going to show these two pathways. One is the transcription coupled nucleotide excision repair pathway, which is known to promote R-loop-induced genome instability. So I tested whether this specific DNA repair mechanism is involved in this repeat contraction. I knocked down two major components. One is called CSB and the other one is called XPG. And both knockdown didn't show any changes in the repeat contraction, indicating this, so it was inconclusive. But and there's another DNA repair pathway that's called mismatch repair pathway, so-called MMR pathway. And this pathway was one of the particularly interesting pathway because this pathway was known to be involved in other repeat expansion or contraction pathway. For example, Huntington disease that are caused by CAG repeat. So I wanted to test one of these player in this mismatch repair pathway, and I knocked down MSH2. And I was able to see very robust and very reproducible suppression of the repeat contraction after this knockdown of MSH2. So based on this observation, I was able to conclude that this R-loop formation recruits this mismatch repair pathway, which in turn triggers CG repeat contraction. So far, I've been shown that this long CG repeat at the Fragile X syndrome cells they are heavily DNA methylated, and this DNA methylation can be removed by 5i or decitabine that I showed later for the neuroprogenitor cells. And once this DNA methylation has been removed, the transcription can happen, which in turn form R-loop. So the nascent RNA can form R-loop. And this R-loop can further trigger stronger DNA demethylation so that it forms positive feedback cycle. So if we start from the dead Cas9 that forms R-loop, it will also cause DNA demethylation, and which in turn will trigger stronger transcription and forming more R-loop. So by this positive feedback cycle, R-loop will be accumulated, and this will recruit the mismatch repair pathway so that this long CG repeat gets contracted, shortened. And this will end up with a shorter CG repeat with unmethylated C, and this FMR1 gene will be permanently reactivated. So based on this observation, I was able to come up with two potential approaches. One is small molecule approach by using 5i or decitabine to remove the DNA methylation. And second method is the dead Cas9 mediated R-loop targeting, so that this R-loop formation can trigger further DNA demethylation and kick out this positive feedback cycle. By using these two methods, we can restore this FMR1 level permanently by achieving these shorter CG repeats. So these approaches can be innovative ways for the Fragile X therapeutics because this is based on reactivating endogenous FMR1 gene. So this is much safer than delivering mRNA for FMR1 to recover this FMR1 level, because that can be much higher than the endogenous level. So if we reactivate the actual FMR1 gene that is in the patient's cells, it should be much safer. And the CG contraction removes the root cause of this disease, and that was caused by the cell intrinsic mechanism, this mismatch repair mechanism. And for this purpose, we use the dead Cas9 that doesn't cut the DNA. So without using exogenous nuclease like regular Cas9, it is much safer than this using Cas9 method. I'd like to thank my PI, Dr. Jeannie Lee, for very long support, and she has been very helpful to progress this project. And I would like to thank Sachiko, Elizabeth, Rodrigo, and Yongwoo for helping me with this project. And I also want to thank Steve Sheridan for helping me to establish this neuroprogenitor cells and give me a lot of advice in terms of the neuronal cell lines. And finally, I want to thank Boyan and Jongmin for very critical discussion and the manuscript discussion. Yeah, and I want to thank Praxa Foundation and Mogam Foundation for the funding. Thank you so much for your attention.